Hello, my name is George Spiro, and I'm introducing the new concept of a virtual reality presentation to illustrate a study that has just been published in the journal Synapse entitled, and you can see at the board, 2 types of somatic spines provide sites for intercellular signaling during calyx of held growth and maturation. And these are the calyces of held, the blue and the green, competing for innervation of the postsynaptic cell depicted in beige. I'll walk you through the key points of the study while exploring selected 3D image volumes used in data analysis. At any time, please feel free to pause the narration, and you do that just by reaching out and grabbing the data. You can resize it and inspect it. We hope you agree that the presentation format is a 21st century solution for efficient communication of science. In order to investigate these somatic processes, we call them spines, and we'll tell you why. At high resolution across developmental age, we utilized serial block-based scanning electron microscopy, which gave us 5 to 12 nanometer pixel dimensions in plane and 40 to 60 nanometer section thickness. We further studied our unique series of image volumes across developmental age and selected those from postnatal days 3, 4, 6, 9, and 30. The calyx has begun to grow on all cells by P4, has reached monoinnervation on 75% by P6 and 90% by P9, which is likely the mature state for this feature. What I have here is a subvolume of these image volumes, which are very rich in information. I'll pull it over here and make it larger. And you can see, for example, synaptic vesicles as the black dots here, mitochondria right here. You can see where membranes are opposed to one another. We don't resolve the individual membranes, but you can see the thicker line where 2 membranes come together. And synaptic sites by the clustering of vesicles near the presynaptic membrane here and here, for example. We'll further explore these image volumes, for example, by cutting through them as I'm doing right now. And you can see many structures and lots of detail and lots of intracellular structures too, and we'll explore that in later scenes. To infer a function from their ultrastructural details. Now, these kind of somatic spines or processes are found on other neurons throughout the brain, not all. Here's an example from the auditory system, the bushy cell in the cochlear nucleus from Canton Moress. The bushy cell actually gives rise to the calyx. It terminates on this principal neuron in the medial nucleus of the trapezoid body. And here's from a study by Sasso et al. Showing the system at postnatal day 9, and the MNTB cell also has a spine-like protrusion. Now let's look at our own material in the reconstruction, blow it up a bit, and you can see now the calyx is translucent, the blue calyx. We can see down to the surface of the cell, and you can see these simple spines sticking up. These are what we call the type 1 spines, and there are many of them underneath the calyx, embedding into the calyx. I want to show you a more complex structure that we noted in our own material and went to the literature, and the only example that we found was here in the ciliary ganglion, where spines, as they're called, were more complex geometrically and intertwined and laid down on the postsynaptic surface into what was called a spine mat. And what we've done here is highlight in magenta these spine mats, and you can get a sense of the complex intertwining of these structures here, and here are several groupings of them, and there are a few more. So in the remaining presentation, we're going to dive into both these structures, the type 1 spines and the spine mats. Pause the presentation and looked around. You've already noted these structures, these very lengthy processes off of the postsynaptic cell that extend quite far out into space. These are very interesting, and these are the subject of a different study. In this next scene, we show selected high-resolution reconstructions of type 1 spines, illustrating their more simple tubular nature, from short at length left up here to longer. In very rare cases, exemplified by the 2 spines at the right, the appearance of a spine head was noted, much like a dendritic mushroom spine. Type 1 spines are typically less than 1/2 micron in diameter and up to a micron in length, similar to dendritic spines, which is part of our rationale for calling them spines. Look to the board, and we show a couple of EM sections through each of these spines that we've reconstructed. And you can see in some cases the spine, like the magenta one, can extend almost to the abneural surface of the calyx. Here we can appreciate the complex 3D geometry of the individual type 2 spines underneath the calyx in different colors, here intertwined into a spine mat. Let's cut through and show their complex geometric relationships to one another. You can see them heavily intertwined. Look to the board and see the same spine mat with the individual spines in exploded view, showing their intricate branching and bending like the spines in the ciliary ganglion spine mats. And hence we use the term spine and call them type 2 to differentiate them from this single simple spines that we call type 1. At right, you see single EM sections through the spine mat, colored to match, and you can appreciate the portions of an astrocyte that is not colored here, that extends deep within the spine mat. So there are lots of locations for pre- and postsynaptic partners in glia to communicate within a spine mat. We've been talking qualitatively about type 1 and type 2 spines, but can we quantify their differences? The way we addressed this was to compute a convex hull by, in essence, dropping a sheet over the structure as seen here in translucent gray, and computing the volume of the convex hull and the reconstructed spine. For the type 2 complex branching structures, in particular, the convex hull volume should be larger than the spine volume, and the type 2 spines are depicted in blue here at postnatal days 3, 6, and 9. If you look to the board, you can see, especially at P6, the volume ratio for type 2 spines, this is the volume ratio on the y-axis, is greater. The blue symbols are above the red in most cases. And note also that many type 2 spines, the blue symbols off to the right here, are larger than any of the type 1 spines. This ratio becomes more similar at P9, but there is a difference here as the type 2 spine begins to collapse upon itself. And there are a few type 1 spines, for example, when they're curved, as in here at P3, where the convex hull then will be larger than the type 1 spine. But in general, we have a metric that differentiates the type 2 from the type 1 spines. Once we defined our type 1 and type 2 spines and spine mats, we looked at their developmental trajectories. Arbitrarily positioned here in space are calyces from postnatal days 3, 6 in the middle, and P30 at the right. That's a mature calyx. At postnatal day 3, and I'll make it a little bit bigger, is shown what is called a protocalyx, when the terminal just begins to expand from a bouton size. Note that type 1 spines in gold are already present. Only in one case did we note a protocalyx without a type 1 spine, indicating the spines are not necessary to begin growth, but that the presence of the spines and calyx growth are very highly correlated. At P6, we see a high density of type 1 spines and a high density of spine mats here in magenta. All the type 2 spines are colored similarly. The spine mats did not appear until the calyces reached a size of about 100 microns squared of directly opposed pre- and postsynaptic surface, which of course we can measure in EM. And then when we look at postnatal day 30, and let's turn it around here and we'll sort of move it, we can see type 1 spines, but they don't cover the entire surface of the calyx, and we don't find any spine mats. Now let's go to the board and look at the population data. You can see the density of type 1 spines in the upper right here grows from postnatal days 3, 4 in orange, and reaches its peak at postnatal day 6, maintains it at postnatal day 9, and then lower than the initial density of postnatal day 3 out at P30. In contrast, the spine mats have a delayed trajectory, so they're not so prevalent at postnatal day 3, not a higher density than much higher density postnatal days 4 and 6, but jumping up at postnatal days 9, and then only in one case did we find them in a calyx at postnatal day 30. Now when we look at the composition of the spines within a spine mat, and this is the distribution of the number of spines within a spine mat, we can see that there are many spines that have many spine mats that have many spines is the one that I showed you earlier, but that most of them have between 4 and 6. These are the modal values here. This is a bin size of 2. And I'll show you one of those a little bit later. We next inquired whether type 1 and type 2 spines were sites for intercellular communication by looking initially for evidence of synaptic contacts onto the spines. Here's a subvolume of our P6 data set, different from the one I showed you earlier. You can see a type 1 spine right here and collections of vesicles at synaptic sites at its base on either side of the spine. Now let's cut through the spine and track it, and you can see it sort of entering into the calyx and disappearing, and now we'll come back. Note also that in addition to having synaptic sites, along its entire length, the membranes are directly opposed to one another, offering another mechanism for communication. We noted also that the interior of type 1 spines are relatively free of stained material, and this kind of stain exclusion zone extends a short distance into the postsynaptic cell before we begin to pick up these subcellular organelles. Look to the board and you can see a couple of other spines where we've outlined them in red, and in dotted lines have depicted this organelle, a kind of staining exclusion zone. The data down here indicate that from their earliest appearance at P3, type 1 spines are innervated, about 70% of them, and that fraction drifts up to over 90% at P30, even though there are fewer type 1 spines. Now let's look at the type 2 spines, and I've cut into the block. The type 1 spine was over to the left here to show the intertwined individual elements of the type 2 spine. This type 2 spine in particular is filled with what appears to be reticulated membrane or other stained material, and you can see an innervation site along the outside of the spine where the spine mat is in communication with the calyx. Most of the synapses are on this so-called outer dome, but they can occur deep within the calyx also. Now let's cut back out of the volume, and you can see where the individual type 2 spines then are connecting with the cell body. Also appreciate the membranes of type 2 spines are also in close apposition with the presynaptic membrane as additional sites for intercellular communication. As we were considering our evidence for intercellular communication via type 1 and type 2 spines, we noted an interesting set of structures. Depicted here are type 1 spines in gold and type 2 spines in magenta for 5 different cells. They almost look like caps over the cells at P9. We noted within the spine mats and depicted in green were elements of the spine mat that were not attached to the postsynaptic cell or indeed any other cell. These were prevalent on 3 of the 5 cells depicted here, and those 3 are shown up at the board in the top panel. Across many spine mats, there were different percentages of spine mat volume that were detached, and I've highlighted a few here with red arrows. And let's zoom in on one right here where there's detached elements in physical contact with the spine mat, but a small percentage of the total. Let's come around to another spine mat here where there are detached elements also in contact with the spine mat, but also some that appear to be drifting up into the interior of the calyx, which is not rendered here, but is obviously located over the spine mat. In the extreme case, and we'll come around here, you can see that the detached elements are now detached from the spine mat. And so across all spine mats there appeared to be a continuum of fractional dissociation, which we take as evidence for a process of transendocytosis of spine mats into the presynaptic terminal. Looking to the board, you can see for these examples I showed individual EM sections of the elements of these dissociated elements extending up into the calyx, and then in this case completely contained within the calyx, which is colored in blue. Let's recap quickly. We provide the first evidence for spine mats in the CNS. Their peak at postnatal A9 is after calyx growth and competition, and may be more tied to later maturation of the calyx and MNTB cell, including fenestration of the calyx. Transendocytosis offers a large range of biochemical communication pathways, and here we have some close-ups now with the beige postsynaptic membrane added, and in this case, in translucent, the blue membrane of the calyx. And you can see this dissociated set of structures here have both membrane and cytoplasmic biochemicals, so a rich set of intercellular communication. Notably, transendocytosis has been described for extrusion of neurospinnules from dendritic spines and hippocampus into the presynaptic terminals, and here's one completely contained. This is from Spacek and Harris. Both type 1 spines and spine mats are also sites for synaptic and membrane-to-membrane direct communication, and type 1 spine prevalence is closely associated with calyx growth. Thank you for your attention. I hope you take the opportunity to pause the presentation and really inspect the data, and consider making your own presentation using this format that we have defined for virtual reality.